So on one side of the paper, what we're going to do is just do a brief overview about cell division. And then on the other side, we're going to look at the nitty-gritty of mitosis. And so on one side here, this part here, is not dealing with the color coding yet. All right. So I just wanted to compare and contrast our two main types of cell division that occur in humans. Think in your head. Can you tell me the two main cell divisions that occur in humans? All right. You should have mitosis and meiosis. Is that what you came up with? All right. Now, this chapter, guys, this chapter is all about mitosis, but I want to compare again meiosis so that we can keep it straight. This is a big picture here. So in mitosis, we start out with a cell, and I'm going to draw the nucleus. Inside that nucleus, how many pieces of DNA do we have if this is a human cell? 46. And just a real quick, when I say pieces of DNA, the reason why I say that is because if a cell is not going through cell division, the DNA is not in what we call a chromosome format. So real quickly, because last hour I got a little bit confused with this, is that remember that DNA are wrapped around these proteins called histones, and if the cell is not going through cell division, it's very loosely packed, and we call that chromatin. And so when a cell is just hanging out before it does cell division, all the DNA is kind of stringy. It is wrapped around these proteins, but it's kind of stringy. Kind of like if I were to hold a thread up here, a white piece of sewing thread, it would be very, very thin. You probably wouldn't be able to see it from the back, and so on. And so very, very thin. And then what happens is when a cell is going to go through either one of these cell divisions, it condenses. What does that mean? It comes together. So the little proteins come together and come together like this. All right, and then you have the DNA still wrapped around it. And so you get a short, stocky little structure that's called a chromosome. All right, kind of like if I were to take that sewing thread analogy and wrap it around a spool, you would be able to see the spool and so on. It'd be noticed, so you'd have a whole bunch of DNA, all right, wrapped close together, and it makes it able to be seen, and so on. And so that's a chromosome. So most of the time, for a cell, the DNA is in this chromatin format. It's very, very stringy and so on. Only condenses when it is going to go through cell division. So that's why I say the cell at the beginning of mitosis, it's really 46 pieces of DNA in this chromatin format, all right? When it goes through cell division, it'll condense into chromosomes, and those chromosomes will rearrange themselves, and at the end of mitosis, another thing, can you answer this? At the end of mitosis, how many cells are we going to have? At the end of mitosis, how many cells are we going to have? What do you think? Raise your hand if you think you know. Peter? It is 2. All right, there are 2 cells at the end. And if I were to draw these cells in the nucleus, what number would I put in there as far as pieces of DNA? 46 in each one of those. All right, 46 pieces of DNA. So mathematically, you might say 46 doesn't equal 46 plus 46. 46, we start out with 46, and we end up with 92, 46 in each one of these. So what must happen to the pieces of DNA? We replicate it. So we make copies. So when we talk about between here and here, the nitty-gritty inside of here is that the DNA would make copies and replicate and then split into 2 different cells so that each new cell is genetically identical. And this cell is not even around anymore because this is split and now it's these 2 cells. All right. And so what kinds of cells? Well, let's put 2 pieces of information here: type of cell and the reason for the cell division. See if you can put that, can you come off the top of your head and write down what kind of cell would go through this type of cell division? and the reason why it would go through that type of cell division. Who wants to take the type of cell? What do you think? Elizabeth? Eukaryotic cells can go through this. So let's go with us. We are made out of eukaryotic cells, so let's be more specific. All of our cells are eukaryotic, which means that they have a nucleus. So which ones or which type of cells in us would go through this? Mark?
Somatic cells.
Somatic cells, which means what?
The body cells.
Yeah, another way to say that. So somatic cells, these are our body cells. So a skin cell, writing skin. And what reason would cells go through this process? Grace?
Repair.
All right, yeah, to repair. So when we cut ourselves, the cells surrounding that cut will divide by mitosis and fill in that area. So we repair ourselves, and what other reason? Yeah. To replace, all right, so we replace, we talked about apoptosis and killing off cells, so then we can replace them, and so on, so that would be true. Kelly? Growth. This is what, when we grow, we become more cells big. That's how we grow. All right? And so, so our cells divide to grow. All right? And that is mitosis. So let's compare this to the process of meiosis. So in meiosis, what's similar to mitosis is that we start out with a similar type of cell. So we have a cell that in us has 46 pieces of DNA at the start of meiosis. These cells are going to make, let's go ahead and, I'm going to leave some space here, and next to here I'm going to write the type of cell. What type of cell is made here? What's the? Yeah, this is the type of cell going through. This is the making of this is the gametes. And so, so gametes are reproductive cells. These are egg and sperm cells. So this is what we're making in meiosis. And so, when this cell goes through division, with this cell, by the way, at the beginning of making of gametes, this cell is found in different places in males and females. So in males, this is the process of making sperm cells, and so that cell at the beginning would be in the testes. And then in females, the females at the end will get egg cells, and this original cell is in the ovaries. So this is where these cells are in the human body. And then they will do this process of meiosis. Meiosis is unique in that it has 2 cell divisions. All right, so I'm going to go here. So what I mean by that is that it divides into 2, and then each one of these 2 divide into 2. So you end up with 4 cells at the end. Now my question to you is, how many chromosomes should those 4 cells at the end have? Ashley? Yeah, they should have 23 chromosomes. And why is that? Or 23 pieces of DNA. What's the purpose for that, Sam?
When during the fertilization of an egg by a sperm cell, the 2 gametes are taking genetic information from 2 parents. So by combining those genes, they need to make the amount of genes that are in a human. They need to match the amount of genes that are in the human, which is 46 genes, and divided by 2, that is 23. So 23 genes in one gamete, 23 genes in the other gamete equals the amount of genes they need.
Yeah, exactly, except instead of genes, we're gonna say pieces of DNA or chromosomes, all right? So yeah, so everybody, we have an egg and a sperm cell, and every fertilization occurs, and everybody starts out as a one-celled baby called a zygote. And the zygote in the nucleus, to be human, has to have 46 chromosomes. All right, so if you have two cells uniting to make a one-celled baby, then these two cells have to have half the number of chromosomes. Otherwise, if you made egg and sperm cells by mitosis, if these were egg and sperm cells, you would have 46 chromosomes in your egg cell, 46 chromosomes in your sperm cell, and the baby would have 92 chromosomes or 92 pieces of DNA. If this happened, if by chance this happened, then the mother would probably never even know she was pregnant because that one cell would not survive, all right? And so therefore it would die and they would never even know that pregnancy occurred. And so for humans, 46 is kind of a magic number in terms of the amount of DNA and the chromosomes that we have. We're going to talk a little bit when we get into genetics that there are some genetic diseases where somebody has 47 chromosomes, so they have one extra one, or 45 chromosomes. But even those, they're real specific about which ones you can have an extra one and which one you can't have an extra one, and so on. And so therefore, it's imperative that we have this separate cell division that reduces the chromosome number by half. Now mathematically here, I'm going to talk a little bit about it as we get into the next chapter with meiosis, but I'm just going to have us put in here right now that there are 23 chromosomes in here, so mathematically 23 when it divides doesn't equal two sets of 23, so just like here, this doesn't equal two sets of 46, so there's some replication that occurs here. So I'm going to put here that right here, these are replicated chromosomes, and this, when you look back at it after we do meiosis, will make more sense. These are replicated chromosomes. So this is what we call meiosis 1. I'm going to put a 1 here, from one cell to two cells. And then we go from 2 cells to 4 cells. This is called meiosis 2. They're considered 2 cell divisions. And these are single or non-replicated chromosomes. All right, and we end up with 23 pieces of DNA. So relating mitosis to meiosis, back to this picture here, we made the egg and sperm cells by meiosis. Fertilization occurs, you get a zygote, and then how does mitosis relate to this?
The zygote will begin to do mitosis and grow.
That's right. This zygote will now begin to do mitosis and divide and grow, and so that one-celled baby will divide into 2 cells, and it'll be 2 cells big, then 4 cells, and so on and so forth. And we'll divide by mitosis, and that's the growing part, all right, that we're talking about. Okay? And so this is an overview of mitosis and meiosis, big picture here. Now, what I want to do is we're going to flip your sheet over and we're going to do and talk about more of the nitty-gritty as far as this part here. So I wanted you to see big picture, what mitosis is, what we end up with, what's the end result, and, without getting bogged down in between the middle part here. And so we're going to draw ourselves a picture that illustrates what I talked about in the notes last night with the pictures from the book, but I think drawing it out and doing some color coding will help people to understand a little bit better. All right? So we're going to flip that over, and I'm going to draw on a big piece of paper right with you so that you can see. Let me grab a piece of paper here. So I'm going to draw up in here. Okay, before a cell actually enters mitosis, and it's just hanging out, because not all cells are dividing all the time, what phase is it in? This phase is actually not part of mitosis. What phase is it in? It starts with I.
The interface.
It's called interphase. All right. So looking at this, look at this here. So what happens you have a cell. I'm going to draw a cell here, and I'm going to draw the nucleus pretty big inside this cell. Normally it's not that big, but I want to draw inside of it, so I want to have enough room to do that. And Interphase, if you recall, has three subphases. They're called the G1, S, and G2. So this is the G1 phase of Interphase. So this is when the cell's hanging out. All right, most cells are in interphase when you see them underneath the microscope, because most cells are not dividing. And so, and I'm going to put here showing one pair. And I'm going to talk about here in just a second what that means. So notice I'm doing the outline and just the writing in black. All right, I'm going to draw the DNA and so on in colors in just a second here. A pair. How many is in a pair? Two, right? So I'm going to draw. I told you I was going to use orange and purple. All right. So I'm going to draw two pieces of DNA. Now the DNA is stringy. All right. So I'm going to draw here one piece, just a squiggly line. It doesn't have to look exactly like my squiggly line. All right. But two pieces of DNA here. This is loosely packed DNA, which means in what form is it in? Chromatin or a chromosome at this time? This is chromatin. And so chromatin, very loosely packed. So if you were to look at a cell under the microscope in this phase, you would see the nucleus, but you would not be able to see these pieces of DNA. Just like it would be hard to see a piece of sewing thread from the front of the room. So let's label this chromatin. So this is chromatin. There's a lot of vocabulary with this. There's chromatin, chromosomes, we'll talk about chromatids in just a second here. All right, let's talk about what it means by showing one pair. All right, I'm going to put this picture up here. Let's say that this is a cartoon representation of pieces of DNA. And what I want to do is have us look at these, all right? And so I'm going to circle this one right here. So notice here, each one looks differently. They're different sizes, shapes, different striations, and so on. I'm going to turn off the lights here. What I want you to tell me is in this field of view, if you can find any matches to that one. Luke found one. Where's it at, Luke?
I thought it was the one in the top left.
Alright, so top left, this one?
Yeah.
Alright, you would be correct. This is a match here. Alright, now that's that field of view. Let's go down here. Any more that match that one? Down here. If you can't find one, you would be correct. There's only one match for this, all right? So that we have two of these kind of chromosomes, all right? Then we could do the same thing with, let's do a different one here. We could do the same thing with this one, all right? Any matches for that one? Grace?
The middle right.
Middle right, right here? Yeah. Yes, all right. And then we could look all around and there would not be another match for that one. All right, we could play this game for a while, but we're not going to. All right, so what my point is that these are 46 pieces of DNA. Two look alike here, two look alike here, and we could find a match for every one. So when we talk about humans, we could say that we have 46 pieces of DNA or 46 chromosomes. Another way to say that is that we have 23 pairs of chromosomes. My question to you is, why are they in pairs? Why are they in pairs? Zach?
What comes from the male gamete and what comes from the female gamete?
That's right. Going back to that picture we drew of egg and sperm cells, where do you get your DNA? You got half of your chromosomes or half of your DNA from your mother, half of them from your dad. And so therefore, every one of these pairs, one came from mom, one came from dad. One came from mom, one came from dad. And so when we say that mom and dad gave us 23 chromosomes, or half of our DNA, it's very specific as far as one of each type of chromosome. All right, so that's what I mean going back to this picture here that we just drew. That's what I mean by showing one pair. All right, so now let's build on this. And so normally for humans we'd have 23 pairs. It would be really messy to try and draw 23 pairs, so we're just going to draw one pair, two pieces of DNA or a pair, and follow it through and assume the rest of it happens to the other 44 chromosomes as well. All right, so right now we're not in chromosome format. So let's go through, and where this cell is going to get ready to divide, what does it have to do to the DNA? It has to replicate it, right? So remember that we want two cells identical to this cell that we started out with at the very end. So in order to do that, we need to replicate our DNA. So we draw our nucleus here, and so for the orange piece of DNA, this is your original piece of DNA. What happens is the cell makes an exact copy of it, and whoops, I don't want to draw it on there. The cell makes an exact copy of it, so this is the original, and the copy kind of looks like this, and it connects to the original at a spot called the centromere. And so this right here is a copy of this side, is a copy of this side. And then the purple, we do the same thing. So here's your original, and we're going to make a copy of it. It's still stringy, so it's still in the chromatin format. All right. So again, if we were to look at the cell underneath the microscope right now, we still would not be able to see very well the pieces of DNA. It may start to condense a little bit. Sometimes at this point you can see like dark little spots within the nucleus, but you can't see individual chromosomes. And so let's write a couple of things underneath. This phase, we're still in interphase, where you replicate it. This is called the S phase of interphase. The S of interphase. S stands for synthesis. Why would they call it that? Because what are you synthesizing? What are you synthesizing here?
A copy of the chromatin.
Yes, absolutely. All right, so that's the S phase of interphase. So I'm just going to put here that the DNA is copied. And I'm going to put here in the G2 phase as well. These are the three phases of interphase. It's kind of hard to draw here, but in our notes you have what happens. It'll have what happens to each one of these phases. One other thing that happens besides the DNA that it is replicated is that the cell grows. So that's kind of it's not reflecting that, but imagine we start out being a zygote. Everybody started out this way. When we divide by mitosis, we were 2 cells and 4 cells and so on. Imagine that if the cell never grew before it divided, your cells would be nothing after a while because it would just keep on reducing in size by half every time a cell division occurs. So it grows before it divides so that doesn't happen. All right, and so this is preparation for cell division. So this is not actually considered part of mitosis. And so because of that, we're going to draw a little line here, right here. I'm going to write here that this is the start of mitosis. I just want to make it clear that interphase was not mitosis. So now we're ready to do mitosis, and we're ready to start that cell division process because we've prepped, we've grown, we've replicated DNA, and so on. So the start of mitosis, I'm going to draw a cell here, start of mitosis. Anybody remember what the first phase of mitosis is? Starts with a T. Mark?
Prophase.
Prophase. Oh, you know what? One other thing. Let's go back back to S phase of interphase, one other thing I forgot to draw in here real quick is that the centrosomes and the centrioles have replicated. So I'm going to go ahead and draw those in here. Draw those in. There's two of them, and they haven't not really doing much yet. All right, but I'll show you in this next picture what they do. So those are centrosomes. Okay, so in prophase, we have a nucleus still intact. So I'm going to draw a nucleus. Notice I left a little space here. I have to draw some things going on with the centrosomes there, so that's why I left a little space here, so you might want to leave some here and then, as well. Let's do what's going on with the chromosomes first. All right, so here, these are still chromatin, and so they become chromosomes here. What does the chromatin have to do to become a chromosome? Condense, all right? They have to squish together and form a shorter, stockier little structure. And this, once a cell does this, you start to see the DNA underneath the microscope. So I'm going to draw my orange one here. I'm going to draw it thicker and stockier and shorter. So this is one side of it. That's one side, and the other side is the replicated part. All right? And where they're touching there is called the centromere. And then I'm going to do the same thing over here with my purple. All right, shorter. And stockier. These are now called what? These are now called chromosomes. All right, so I'm going to just put C-zone. That's my own little shorthand. You can write it out or adopt your own or use mine. Shorthand there. But those are now chromosomes. So that's what happens there with the condensing. So that's why we say in general we have 46 chromosomes. So this is one pair of chromosomes. All right? And then what's going to happen, remember big picture, we're going to want two cells at the end identical. And so these centrosomes, what happens is, I'm going to draw these guys out here. I'm going to draw one right here. And then I'm going to draw the other one over here, so they're separating, kind of to opposite sides of the cell. They're not quite to total opposite sides of the cell yet. Anybody know what grows from these? Why these are important to cell division? What grows from these, Matt? Microtubules, and these are going to form what's called the spindle. And the spindle is going to be helpful in separating the chromosomes properly in mitosis. And so when, at this point here, when the chromosomes here are able to be seen because they've condensed and so on, I want to just talk a little bit about what you can do. So you can see it underneath the microscope, and we're going to be doing a lab with this where we're going to look at cells going in different phases of mitosis and so on. So you can see it. You can't see really well, but you can see dark spots in the nucleus. But once the chromosomes condense in cell division, what happens is that in order to see them and to study, like for instance, to see if somebody has the correct number of chromosomes and so on, what they would do is they take a cell in the process of mitosis and where the chromosomes have condensed so you can see them better. And they take that cell from the person that they want to see, you know, if they have an extra chromosome or something like that for genetic testing or something. They take that cell, and this is a blast from the past, they put it in a hypotonic solution. Anybody remember what hypotonic solution means? What's the cell going to do if it's in a hypotonic solution? Here's the cell. Outside is hypotonic. What does that mean in terms of the outside and concentration of water? Is there a higher or lower concentration of water outside of the cell than inside the cell? Peter?
Lower.
And where?
Outside the cell.
Opposite. Higher. All right, is higher. Higher concentration outside the cell. And so therefore water will go in. What's going to happen to the cell? It's gonna balloon up, right? They don't let it explode. All right, so what they do is then they take it and they suck it up like in a little micro pipette, and they hold it high up above a microscope slide, and then they squeeze and let it free fall to the microscope slide. What's gonna happen when it hits? Split, right? Including the nucleus and all the chromosomes, split out. Then they take a dye that will adhere to the chromosomes themselves, to the DNA, so they dye the chromosomes so that they can see them. And on this microscope that they use, they have a camera on it that's connected to a computer, so they can see all of this on computer. So now they have these chromosomes splattered across the screen. And then what they do is they not only count them to see if they have 46, but they actually match them up in pairs. So remember, there were pairs of them, one that you get from mom, one that you get from dad, and they can actually do this on the computer screen where they can move the chromosomes around, and they match them up with their pairs, and what you end up getting is what's called a karyotype. Do you guys remember this from bio? A picture that looks like this, where you line up your chromosomes, largest to smallest in pairs. Does this look familiar? Yeah. All right. And so the reason these are replicated chromosomes, the reason why is to make a karyotype. In order to see the chromosomes, it has to be where the chromosomes have replicated and condensed into chromosomes, so you can actually see them. And so then what they do, you probably in bio did a karyotype lab. Did you guys have to cut it out and match it up?
Yeah.
That's very popular for bio class. All right, so I'm not going to make you do that. You've already had that experience. And so here's a karyotype. And so every chromosome, largest to smallest, every chromosome is different in its size and in its striation and its pattern. But I just want to point out here that when you're looking at this, that this is one chromosome. This is a replicated chromosome. So this side is an exact copy of this side. So that's like what we just looked at here, exact copy of the other side. And this is the other replicated chromosome. This side is an exact copy of this side, and this would be this chromosome here. So when we're looking at one pair there, that's what we're talking about here. So then they match them up, and that way, usually people only have this done if it's suspected that the person may have additional or missing chromosomes. And so therefore, by matching them all up, they might find that they have an extra chromosome or a missing chromosome. Notice the only thing that doesn't follow the rule is on the bottom right here, the sex chromosomes. Sex chromosomes are called that because they determine sex or gender of the person. And so there are two in humans, two sex chromosomes, an X and a Y. Notice that for this one here, there's two X chromosomes and no Y chromosome. People are going to have almost always two sex chromosomes. This person has two X's. That is for a male or female, do you know? This is a female. And how would the males look different? They have a Y. And so males have an X and a Y. Notice that they don't match. All right, the X and the Y, but these are still considered a pair, and the combination of one X and one Y determines maleness. So these guys don't have similar genes on them. The Y chromosome on the DNA has very different genes than the X chromosome, but that makes a male. And then females, for the two X chromosomes, they have similar genes. What do I mean? What makes these guys pairs, and why do they look alike? It's because that if you look at the DNA for one of these here, so if you looked at this DNA, you might have, this is the DNA here. From here to here might be a gene. So I'd say, okay, let's say right here is a gene, all right? That means this segment of the DNA here. This gene, and since this is an exact copy of it, will be the exact same gene, and this might be a gene for eye color. What that means is on the second X chromosome in that same spot, there's also a gene for eye color. So that way, anybody who has an X chromosome, even males for humans, in that same spot on their X chromosome, they have a gene for eye color. And so it doesn't mean that you have the exact same gene for eye color. So there's brown color, there's green color, and so on, but there's an eye color gene there. And so therefore, these matches not only look alike, but the types of genes on those chromosomes are also the same types of genes. All right, you can have different versions of those genes, but they're the same type to determine the same trait. All right, And so, so that's your karyotype. And so we are looking at, and so when you make this, we're looking at cells that are going through mitosis and have condensed like this, all right? So let's go back to the process of mitosis and look at what happens next. So let's draw, we're gonna draw just so for spacing purposes here. I was gonna fit 2 more cells on here, all right, for 2. If you can't, there'll be enough room down here to put it, but I like to show people spacing. All right, here's another cell here, all right, and anybody know what the next phase is? And in our textbook, it talks about prometaphase. This happens before metaphase. And you have to remember that it's not snapshots like this happens, and this happens, and this happens. Like, it happens in this, but it's really like a movie, and we're taking like screenshots of the movie at various points. All right, so prometaphase. Let's draw in what happens here. So now the centrosomes are on opposite sides of the cell. So let's do that first. All right, let's draw 2 little squares here with these little guys protruding out, opposite sides of the cell. All right, the chromosomes are going to start to move towards the middle of the cell. Metaphase, which is the next phase, means middle, but they're not quite there yet. So, so I'm going to draw, all right, like let's say here, so moving towards the middle, here's my orange one. Moving towards the middle, not quite at the middle, and same thing with the purple, all right, let's say right here, moving towards, but not quite at the middle. And it's still, these still are enclosed in a pseudonucleus. What's going on is this nucleus is going to break down. So from here to here, the nucleus breaks down. So I'm going to go ahead and draw to show that. I'm going to draw it like this. All right, so the nuclear envelope, like a dotted line to show that it's breaking down. And then, coming out of the centrosomes here are those microtubules. You have some microtubules that actually attach to the centrosome of each one of these chromosomes. So I'm going to draw one right here to this chromosome, and another one from this side to this. So therefore, each chromosome has 2 microtubules attaching to it, one from both sides. Now, some microtubules, their job is to attach to the chromosomes. Other microtubules, their job is to elongate the cell. So I'm going to draw here 2 that kind of overlap like this, 2 that overlap like this. What they're going to do is they're growing and going to elongate the cell this way, because what we're going to end up doing is splitting the cell in half. So that's what their job is. So there's 2 different types of microtubules there. And this is where I want to stop here for the day. So we'll finish this up tomorrow. I do want you to watch part 2 of the notes, and then if you would bring up any colored pencils that you borrowed from me, a computer, put it in the correct bin, I would appreciate it.